Hi everyone, in this video I will be explaining you cell cycle and its regulation. So the average duration of the cell cycle is around 18 to 24 hours. There are two phases for cell cycle. One is the M phase or the mitotic phase. So M phase is the phase where the actual cell division takes place, and the remaining phase is together known as the interphase. So during interphase the cell prepare itself for the cell division. So before going for the cell division, cell needs sufficient amount of RNA, DNA, protein and cytoplasm. So all these requirements are attained during this interphase. And this interphase again divided into three phases. One is G1 phase, S phase and G2 phase. So during this preparative stage the cell prepare for a cell division, and during M phase the cell division occurs. And there is one more phase that is known as G0 phase, that is the dormant phase. So in a cell population most of the cells are in dormant phase. So let's see the events takes place during each of these phases. The first one is the G1 phase. So protein synthesis and RNA synthesis takes place during G1 phase of the cell cycle, and the average duration of this G1 phase is around 6 to 12 hours. And DNA synthesis, that is the DNA replication, that takes place during S phase, and the average duration of the S phase is around 6 to 8 hours. Once the DNA are synthesized, there will be a chance for DNA damage, so that should be repaired by some DNA repair mechanism. So that DNA repair takes place during G2 phase, and there will be condensation of DNA into their corresponding chromosome form and cytoplasmic enlargement. All this takes place during the G2 phase of the cell cycle. And regarding M phase, that is the mitotic phase where the actual cell division takes place. So during mitotic phase the cell divides into two daughter cells, and it is the shortest phase of the cell cycle, and the average duration is around 1 hour. And after the M phase the daughter cells are either enters into a dormant phase or re-enter into the cell cycle. And in a normal cell population most of the cells are in G0 phase, that is the dormant phase. So these are the phases of cell cycle. M phase is the phase where the actual cell division takes place, and the remaining phases are together known as the interphase. So interphase contain G1 phase where the RNA and protein synthesis takes place, and S phase where the DNA synthesis takes place, and G2 phase where the DNA repair mechanism, also the cytoplasmic enlargement. These are the events takes place during G2 phase. And once the cell has all the requirement for a cell division, then the cell enters into the mitotic phase where a cell is divided into two daughter cells. And after the mitotic phase the cell having two phases, either they enter into G0 phase, that is the dormant phase, or they enter into the cell cycle. Coming to the regulation of cell cycle, there are a number of checkpoints to regulate this cell cycle. There are three checkpoints. One is in the G1S interphase, the second one is in the S phase, and third checkpoint is in the G2M interphase. And these cell cycles are regulated by a specific proteins, and these proteins are known as cyclin. So cyclins are cell cycle regulatory proteins, and there are four different types of cyclin protein: cyclin A, B, D and E. And this cyclin are active only in presence of a phosphorylating enzyme, that is kinase group of enzyme, and that cyclin dependent enzymes are known as cyclin dependent kinase, because these phosphorylating enzymes are active only in presence of cyclin. That's why they are known as cyclin dependent kinase. There are 5 different types of cyclin dependent kinase: cyclin dependent kinase 1, 2, 4, 5 and 6. So cyclin protein activate the cyclin dependent kinase enzyme, and this activated kinase enzyme phosphorylate other regulatory proteins of the cell cycle. So there are two important cell cycle regulatory protein. One is retinoblastoma protein, and the other one is P53 protein. These are regulated by the cyclin and cyclin dependent kinase enzyme. So cyclin protein activate the cyclin dependent kinase enzyme, and once the kinase enzymes are activated, that this activated kinase enzyme phosphorylates the specific regulatory proteins of the cell cycle. So let's see one by one. The first one is regulation by retinoblastoma protein. So retinoblastoma protein, it is named because this protein was first isolated from a patient with retinoblastoma. So retinoblastoma protein is a cell cycle regulatory protein. So what is the function of this retinoblastoma protein? It inhibits the cell cycle at G1 phase. So normally this retinoblastoma protein inhibits cell cycle at G1 phase. So whenever cell need more cell, that is whenever cell needs cell division, this retinoblastoma protein has to be inactivated, and this is done by cyclin dependent kinase enzyme. So cyclin activates cyclin dependent kinase enzyme, and this activated cyclin dependent kinase enzyme phosphorylate this retinoblastoma protein. Once it is phosphorylated, it becomes inactive. So phosphorylated form of retinoblastoma protein is inactive in nature. So they can't inhibit cell cycle. So cell proceed for the mitotic phase. That is the normal events takes place during cell cycle. But there are some cancers causing viruses like herpes simplex virus or human papilloma virus. These types of tumor antigen permanently inactivate this retinoblastoma protein, so that will lead to uncontrolled cell proliferation and tumor formation. So normally this retinoblastoma protein inhibits cell cycle at G1 phase. So whenever cell needs the cell division, these retinoblastoma proteins are temporarily inactivated by cyclin dependent kinase enzyme, and these phosphorylated retinoblastoma proteins are inactive form. But in case of some tumor causing viruses, especially herpes simplex virus and human papilloma virus, this tumor antigen permanently inactivate the retinoblastoma protein and produce uncontrolled cell proliferation. And let's see the cell cycle regulation by p53 protein. So p53 protein, it is also a cell cycle regulatory protein, and these proteins are activated whenever there is a DNA damage. So what is the action of this cell p53 protein? They also inhibit the cell cycle at S phase. So whenever there is a DNA damage, these p53 proteins are activated, and this activated p53 protein having two functions. One is they temporarily arrest the cell cycle, and they promote the DNA repair mechanism. So once the DNA damage has been repaired, the cell enters into M phase. If there is a severe DNA damage, then the cell enters into the apoptosis. Apoptosis means a programmed cell death. That is a normal biochemical event takes place in cell population. So whenever there is a DNA damage, these p53 proteins are activated. So what is the role of p53 protein? They temporarily inhibit the cell cycle, and they promote the DNA repair mechanism. Once all these damages are repaired, then the cell proceed for the cell cycle. And if there is a severe DNA damage, then this p53 protein promote apoptosis, that is the programmed cell death. So p53 protein play an important role in maintaining genetic stability. So a few points regarding p53 gene, that is the gene coding for p53 protein. So the role of p53 protein is it is a cell cycle inhibitor. So p53 gene is a tumor suppression gene because they prevent cell division. And this p53 gene play an important role in multicellular organism for the prevention of cancer. And p53 gene has been described as the guardian of the genome because they have an important role in preventing genetic mutation. And the p53 gene is located on the short arm of the 70th chromosome, and it is named as p53. This is because of its molecular mass. The molecular mass of p53 protein is around 53 kilodalton fraction of the cell proteins. So in a normal cell population, cell cycle is a well-regulated event, and these are regulated by different types of regulatory proteins like cyclin-dependent kinase enzyme, retinoblastoma protein, and p53 protein. So in a normal cell, the cell cycle is a well-regulated process, and most of the cells are in dormant phase. Only 1% of the cells are in the mitotic phase. But in case of a cancer cell, all these regulatory processes are disrupted, so that will result in uncontrolled proliferation of the cell and cancer development. So that's all regarding cell cycle and its regulation. Thank you.